Hello everyone, good evening and welcome to Practically's NEET Bio class. So today we're going to learn about the eukaryotic cell. In the previous classes we were learning so many things about the cell. We learned about the cell theory. We learned about the prokaryotic cell, which is the primitive kind of cell. We learned that how prokaryotic cells don't usually have a very developed structure. They have very few organelles and they have a false nucleus. They don't have a true nucleus. Okay. So today we're going to learn about the eukaryotic cell, which is actually the developed cell. Okay. So let's get started. Now, first, I'd like to talk about Practically. Practically is India's first experiential app. You get a lot of new features every single day. There's something being added in the app. It is very interesting as well as educational. It runs for 6 to 12. You can also follow us on our socials in Facebook and on Instagram. And don't forget to like, share, and subscribe to our YouTube channel. Okay, so eukaryotic cells. Now, eukaryotic cells are actually the developed cells. Okay, so what do I mean about developed is the foremost thing you have to think when you talk about eukaryotic cells is they have a true nucleus. They have a proper nucleus. Okay, that is one of the most important things when it comes to eukaryotic cells. Okay, because the prokaryotic cells don't usually have a true nucleus. They don't have proper covering. It's just some kind of genetic material floating in the cytoplasm. Okay, so when it comes to eukaryotic cells, they have a proper nucleus. They have a covering. There is nucleoplasm. There is a like a proper nucleolus and everything. Okay, apart from that, there are n number of organelles which are present in eukaryotic cells. Okay, because in prokaryotic cells we did not have like proper organelles. There was just ribosomes, that's all, and some cytoplasmic things. There was nothing else. But here you can find a lot of organelles. You can find mitochondria, lysosomes, vacuoles, chloroplasts. Okay, so different kind of cells have different kind of organelles. Okay, so that's why it is a developed cell. Okay, developed cell. Now, where do we find these eukaryotic cells? We usually find these eukaryotic cells like anywhere except bacteria, blue-green algae and all. We find it in protistas, we find it in plants, animals, fungi, each and everything, be it unicellular or multicellular, they are all eukaryotes. Even we have eukaryotic cell. Okay, so they have a proper cytoplasm and they have a number of membrane-bound organelles. Okay, apart from that, they have a properly organized nucleus. With the nuclear envelope, there is also a covering. Okay, because we saw that in prokaryotes, it was just some genetic material floating in the cytoplasm, no cover, no nothing. Okay, here there's actually an envelope which actually protects the nucleic materials and everything. Okay, so this is why it is a true nucleus. Okay, and apart from that, they have proper cytoskeletal structures which gives the cells a bit of structure. They have vacuoles, chloroplasts, mitochondria and everything. Okay, and on the other hand, like plants have some kinds of extra cell organelles. Like plants have big vacuole. Okay, they have a chloroplast. Okay, they also have a cell wall, which is actually absent in animal cells. On the other hand, animal cells have proper lysosomes. And they also have centrioles. Okay, this and all are not present in the plant cells. Okay, so we can see how developed eukaryotic cell is. When I was talking about prokaryotes, all I talked about was the cell wall, a bit of cell wall, a bit of cell membrane, a false nucleus, some ribosomes, that's all. But when it comes to eukaryotic cells, so many things are there, one after the other. There are n number of things which are there in the eukaryotic cells. Okay, so let's see who's here. Hello, Vidushi. Welcome to the class. If you have any doubts, please leave it on the chat box. Okay. Okay. So this is how a plant cell and an animal cell looks. Okay. So this is the plant cell. Okay. And this is the animal cell. Now, animal cell, you might see it is mostly oval in structure, and it is oval and kind of irregular. Here, plant is almost a kind of rectangular. It is kind of almost rectangular. Almost, I'm saying. Okay, it's not a proper rectangular. It is almost rectangular in nature. Plus, you can actually see there is a big vacuole over here in plant cell. They have chloroplasts and everything. Here, on the other hand, you can actually find centrioles and lysosomes. See here, you can find some lysosomes and all you can find. Okay, so you can find a lot of things, different things in the plant and animal cell. Okay, some things will be there in plant cell, some things will be there in animal cell. Okay. So this is how basically a eukaryotic cell looks. Eukaryotic cell can be a plant cell also. It can be a animal cell also. Okay. So first, we'll start talking about the different parts in the eukaryotic cell. Today we'll just, since it is so huge and there are such number of things in the eukaryotic cell, so I have divided it into two classes, okay, two parts. First we'll discuss today we'll be discussing some of the organelles. In the next class we'll be discussing some of the organelles, okay. So first is the cell wall. Now cell wall, be very sure that cell wall is not present in animal cells. Okay, so they are not present in animal cells. They are present in plant cells, fungi, okay, algae and all. They are present, but not in the animal cells. They are not present. Okay, so what is it? It is a tough, rigid layer that surrounds some of the cells. Okay, it's very tough and rigid. Okay, and it is located right outside the cell membrane. So animal cells, they directly have the cell membrane, they don't have the cell wall. But here in plant cells and all, you can actually find the cell membrane, and their function is to actually provide rigidity, strength, protection, from mechanical stress and infection and everything. See, try and understand this. When we talk about plants, we know they cannot move. Okay, so they need some strength in their cells. The whole body is made up of cells, right? So they need some kind of strength to actually stand in one place, to actually protect themselves from different kinds of weathers, different kinds of environments. So this kind of strength, this kind of protection can only come from something like cell wall. Okay, cell membrane is there, but they have to secure themselves, right? So that kind of strength comes from the cell wall. Okay, so the cell wall is a characteristic feature to cells of plants, bacteria, fungi and algae. Protozoans and animals, they don't have a cell wall. As I said, they don't have protozoans and animals, they don't have a cell wall. And the compositions in the cell wall, they are a bit different in different things. Okay, so like in fungi cells, this is a cell wall in fungi cells. Cell wall in fungi. Okay, so what they have, they are basically made up of glucosamine and chitin. So chitin is a very strong thing. Okay, chitin is actually a very strong material. Apart from that, they have glucosamines, as in they have carbohydrate products also, and they have proteins also. Okay, so they have beta glucans. Okay, see, these gray structures are beta glucans, and on top of that, they have some kind of protein structures called manoproteins. So everything, whatever fungi has, they have chitin, they have beta glucans, plus they have some manoproteins. So these are all three of them are quite together. They are quite strong and rigid, and they provide a proper protection. Okay, so this is in fungi. In plant cell, again, it is composed of cellulose, hemicellulose, glycoproteins, pectins, and lignin. So in plant cells, there are different kinds of things because the plant cell wall can actually be divided into structures, different structures. Okay, primary, secondary, middle lamella, like that. So, and each and every one of them are different. Okay, and cell wall are basically non-living nature. Cell wall do not live. They are not living cells. They are dead cells. Okay. Now, the plant cell wall. Okay, we were talking about the fungi. We were talking about the fungus cell wall. Now we'll talk about the plant cell wall. Okay, so the plant cell wall consists of basically 3 layers. Okay, so there is primary cell wall, there is secondary cell wall, and there is middle lamella. Okay, so this is basically how it looks. Okay, the cell wall, this is how it looks. There is a primary cell wall. This is the primary part. Okay, this is the secondary part. These are insides, okay, plasma membrane. And somewhere in the middle here is the middle lamella. Okay, so the middle lamella is the first thing. It is outside, it is right outside. So it is the first layer formed during the cell division. The layer is rich in pectin, and the outermost layer joins together the adjacent plant cells. So what the middle lamella does is it just puts the different things together, different cells together. Their work is to keep the cells together. Okay, different plant cells, they have to keep themselves together with the help of middle lamella. Now, after that, it is the primary cell wall, and it is made up of pectin, hemicellulose, and glycoproteins. So that is how. So the primary cell wall is made up of pectin, hemicellulose, and glycoproteins. Okay, and it is like a framework. It is kind of a matrix type of a area. After that is the secondary cell wall. Now this is the thicker layer. See, here you can actually see it is a thick layer. Primary cell wall is actually very thin. Okay, here you can actually see that the secondary cell wall is the thicker layer, and it is extremely rigid. This is the one, the secondary cell wall is the one which provides the rigidity. Okay, it is composed of cellulose and hemicellulose and lignin. So it's because there is cellulose and hemicellulose, lignin, these kind of things, lignin, cellulose, these kind of things actually provide a lot of rigidity. They are quite strong in nature. Okay, and they provide a lot of rigidity, the strength, the rigidity, the strength to actually face odd kind of temperatures, odd weathers, okay, mechanical stress and all. This kind of strength is provided by the secondary cell wall. Okay, now what are the cell wall functions apart from the strength and everything? So basically they give the shape. Okay, why? Because see, as I said, the plant cell wall actually has a proper shape. They are kind of rectangular in nature. Okay, but when it comes to the animal cell, it does not. It's kind of ovalish and does not have a proper. It's irregular. So that kind of shape, because the cell wall is there, that is why there is a definite shape and structure in the plant cell. Apart from that, there is structural support. there is protection against, as I said, infection, mechanical stress. It also enables the transport of materials from one part to another. They have a thing, see, they have in between two cells, see, if this is a cell and this is a cell, they have a tube-like structure. Okay, so this tube-like structure actually helps in translocation and communication. So this tube-like structure is known as plasmodesmata. So it is like a tube-like structure. In each of the cell, will be joining the cells, okay, for the translocation of cell molecules or communication with each other. That kind of cell, that kind of structure is known as plasmodesmata. Okay. Now, cell walls also help in osmotic regulation. It prevents water loss. It helps in the biochemical activities and prevents the cell from rupturing due to pressure, turgor pressure and all. Okay. The strength and the capability to handle stress and gases exchange and everything is done by the cell wall. Okay, so basically it is protection and communication that is done by the cell wall. Protection and communication. Okay, so let's see. Okay, Shiva, can you just go out and then come back to the video again? Maybe the presentation will be clear for you. Okay. Now we'll discuss about the cell membrane. So this is where the cell membrane is from where the animal cells and the protozoan cells, they start. Okay, so this is present in everything, all of the living beings like plants, animals, fungi, even bacteria. Okay, all of the things, they have cell membrane. Okay, so it is the boundary which separates the living cell from its non-living surroundings in most of the cells. In most cells. Okay, so in animal cells, plasma membrane is present in the outermost layer of the cell, but in plant cell, as we know, it's the cell wall. So this is the outermost layer in animal cell. Okay, it is flexible, okay, because of its structure. Okay, it is flexible and a living membrane, because cell wall was actually dead. Okay, just now I told you cell wall was actually dead. Cell membrane is a living structure. Okay, and it's composed of basically two main layers. They have other things also, but they have two main layers, that is the phospholipids and the proteins. Okay, and it is semi-permeable. Okay, only selected molecules only they'll allow. Selected molecules can pass. Okay, and there have actually been a number of theories regarding the cell membrane. Okay, because cell membrane is actually kind of a complex part of the cell. Okay, so people from just after they discovered that there is something called cell membrane, people had a number of theories according to it. What it is made up of, what are the different parts of the cell membrane. Okay, so a lot of people gave a lot of structures. Okay, one of them was actually PLP, protein lipid protein. Okay, that is not a true model, but some scientists actually say that PLP, that is protein lipid protein. Okay, so people were talking like how they have a protein layer, then they have a lipid layer, then again they have a protein layer, something like that. But finally, scientists known as Singer and Nicholson, they actually found out how it looks, and that is, there are some modifications still again, but the fluid mosaic model is the one still actually people talk about. Okay, it's the fluid mosaic model. People actually, like, when people talk about cell membrane, they feel like the fluid mosaic model is the closest model which comes to the cell membrane. So what is this fluid mosaic model? Fluid mosaic model looks somewhat like this. Okay, so what it is, the first thing the fluid mosaic model actually says is that the cell membrane consists of a lot of, it's not only proteins and lipids, there are actually a lot of things embedded in the fluid mosaic model, in the cell membrane. So first is the phospholipid. Okay, this is the most important part, that is the phospholipid. Okay, apart from that, there are some cholesterols. Proteins are also quite important. Okay, and there are some carbohydrates. So the two important part in the like cell membrane is the phospholipid and the proteins. Apart from that, there are cholesterol and carbohydrates, but that is just embedded in the structure. Okay, cholesterols and carbohydrates are only embedded, just little they are here and there, they are embedded in the structure. But the two main things which actually form the cell membrane is the phospholipid and the protein. So how? Now, the first thing you have to know is plasma membrane thickness is actually a bit thin. It's around 5 to 10 nanometers in thickness. So the main fabric that we are talking about, that is the phospholipid, is it's kind of dual in nature. So this is how it looks. So this is how a phospholipid molecule looks. So this is the hydrophilic part. Hydrophilic as in it loves water. It is not repelled by water. It is attracted to water. So this is the hydrophilic part. And this tail structure is actually hydrophobic. Okay, so how is it placed in the membrane? It is placed somewhat like this. Okay, so it is kind of a double layer of phospholipid. Okay, so the hydrophobic portions are inside and the hydrophilic portions are outside. So this is the phospholipid. The other thing is the protein. Okay, now protein, you can see here, proteins are embedded. Since there are phospholipids all over, the main structure is phospholipid. It is all over the cell membrane. These proteins are embedded. Okay, embedded inside the membrane. And that is why, since they are embedded, integrated inside the membrane, they are known as integrated proteins. Okay, now there are, as I said, there are some glycoproteins, glycolipids, just little there are. These are not the important things, lipids with carbohydrates, proteins with carbohydrates. There are some portions, okay, embedded, just little they are embedded in the plasma membrane. But the main portion are these orange colored phospholipids and these blue colored proteins. Okay, so this is how the cell membrane looks. Now, what are the functions? Okay, so it is kind of a very important thing cell membrane is. So they have a lot of functions, a lot of different functions are there. Okay, so the first thing is they go through a lot of different like functions such as intercellular junction, secretion, endocytosis, cell division, cell growth. Okay, so different kinds of functions they help in. That is, they provide support. But what they actually do is since they're semi-permeable, they'll be giving up molecules, taking in molecules and all that. So that is one of their important functions. Okay. So many molecules can move briefly across the membrane without requirement of energy. That is called passive transport. So when there is no energy required and a molecule just easily they pass through the membrane, that is the passive transport. Okay, see here, you can see there is some molecule and it just crosses. Okay, it just crosses the membrane. So this is passive transport. There is no energy required, there is no push required for it. So it is passive transport. Now, neutral solutes may move across the membrane by process of simple diffusion. Diffusion also takes place. Okay, just like that, they pass neutral solutes, molecules, they'll pass. Water also goes from a region of, if there is water, there is less water inside, they'll go from higher concentration to lower concentration. Or if it's high, vice versa, then also they can change. Okay, so that water diffusion is called osmosis. So passive transport, diffusion, osmosis. Okay, sometimes the molecules cannot pass. Okay, and they need an extra energy push. Okay, when there is an extra energy push from there, okay, energy in the form of ATP, that is adenosine triphosphate. Okay, that kind of push, okay, and that kind of transport of materials when there is an energy push, that is known as active transport. Okay, see here in this picture, you can actually see here there is a molecule and it wants to pass. Okay, but maybe it cannot. So there is an energy over here and energy is being produced. So the molecule then successfully, due to the generation of energy by ATP, they pass. So this is active transport. So there are different things such as active passive transport, diffusion, osmosis. So a lot of things are handled by the cell membrane. Okay. Now after this, we'll discuss about the endomembrane system. Today we'll discuss till the endomembrane system. There are around 4 to 5 organelles under this. Okay. In the next classes, we'll be discussing about the double membrane bound organelles such as chloroplasts, mitochondria, and etc. Nucleus. Okay. So let's see if there are any questions. Okay, so now endomembrane system, why is it known as endomembrane system? Because they help in the transfer of molecules from one part of the cell to other, or from inside to outside and everything. So those kind of organelles which help in that transfer and everything, they are known as the endomembrane system. Okay. This is an important question for NEET. It has actually come where they ask what are the organelles in the endomembrane system. It is endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Okay, so these are the endomembrane system. Now, what are not there, since functions of mitochondria, chloroplast, peroxisomes are not coordinated. They are not a part of putting molecules out or making molecules out or synthesizing molecules and then putting them out and all. They are not a part of that. That is why they are not in the endomembrane system. It is just endoplasmic reticulum, that is ER. It can be smooth endoplasmic reticulum also, rough endoplasmic reticulum also. Then there is Golgi complex. Okay, there is lysosomes and vacuoles. Okay, be very sure that this lysosomes is mostly present in animal cell and big kind of vacuoles, not small vacuoles are present in animal cells, but the bigger ones are present in plant cell. Okay, so first we'll learn about the endoplasmic reticulum. Endoplasmic reticulum is basically surrounds the nucleus. Okay, in a cell, this is how they surround the nucleus. If this is the nucleus, this is this green color portion is the endoplasmic reticulum. So it just surrounds the nucleus. Okay. So these are actually very small structures, tiny tubular structures scattered in the cytoplasm. Okay, and they have two distinct compartments. They have an inside part, that is the luminar part, and the external part, that is the extraluminal part. And sometimes the endoplasmic reticulum, okay, can be divided into rough endoplasmic reticulum and smooth endoplasmic reticulum. Why like that? Because in the rough endoplasmic reticulum, there are ribosomes attached to it. Okay, ribosomes are attached. So since there are ribosomes attached, if you touch it, it will be rough in nature. Here in this picture, you can see there are these red markings of the ribosomes. Okay, so there are ribosomes attached to it. That's why they are known as rough endoplasmic reticulum. And when they are not there, see here, there are no ribosomes attached. So they are known as smooth endoplasmic reticulum. Okay, and rough endoplasmic reticulum is frequently observed to actually help in the protein synthesis, because ribosomes help in protein synthesis. So they have a support function. Okay, and smooth endoplasmic reticulum helps in lipid synthesis. So rough endoplasmic reticulum, because they have ribosomes attached, that is used for protein synthesis, and smooth endoplasmic is used for lipid synthesis. Okay, that is the endoplasmic reticulum. Now comes the Golgi apparatus. Now, 1 second. Okay, so now the Golgi apparatus was actually discovered by this person called Camillo Golgi, okay, in the year of 1898, and the Golgi apparatus came from there only. The name Golgi apparatus came from there. Okay, Camillo Golgi. And what is Golgi apparatus? They are basically flat sacs, okay, disc-like sacs, and it's very small, 0.5 micrometer to 1 micrometer diameter they have. So they're very small, flat, sac-like structures, okay, and they're just stacked parallelly on one top of like this. They're just stacked like pancakes. They're stacked on one top of each other. Okay, and these stacked things are known as cisternae. Okay, these stacked sac-like structures, they are known as cisternae, and together it is known as the Golgi apparatus. Okay, so what they have, the Golgi cisternae are concentrically arranged near the nucleus with distinct convexes or the forming phase and concave trans or the maturing phase. So they have two parts. They have a cis phase. Okay, so they have a convex like this structure they have, that is the cis phase. Okay, this is forming. Okay, and there is also a trans phase. Okay, that is the maturing. So they have two phases. Once on the other one side there is the cis phase, that is the forming phase, and one side there is the trans phase, that is the maturing phase. Now, these are actually not connected with each other. Okay, they are entirely different, but they are interconnected. Okay, their work is also different, but they are interconnected with each other. So what happens is the Golgi apparatus principally they function as packing materials, okay, to be delivered either to the intracellular targets or excreted outside. So what they do is they pack materials, whatever there is formed, okay, whatever that is being formed in the cell, they pack it. Okay, they pack it in a way. See here, they are packing things. Okay, they take it, transport it, and they pack it in a way so that it can be transported from one part of the cell to other, or they can be transported to the outside of the cell. Okay, so they have a packaging function. Okay, materials to be packaged in the form of vesicles from ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. So as I said, there is the cis phase and there is the trans phase. So materials which want to be packed and want to be sent out, such as proteins, lipids, whatever it is, they come to the cis phase, the forming phase. Okay, and they come inside and then they start, since they are interconnected, they start traveling towards the trans phase. Okay, and that is why it is so associated to endoplasmic reticulum, because endoplasmic reticulum is sending things. No, it is sending proteins, it is sending lipids. So these proteins and lipids are getting packed in the Golgi bodies and getting pushed out or pushed somewhere else. Okay, a number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae and they are released from the trans. Just now I said proteins, lipids, okay, everything gets packed from the cis phase to the trans phase and goes off. Okay, that is the function of the Golgi apparatus. Now comes the lysosomes. Now lysosomes basically are found in the animal cells. Mostly they are found in the animal cells. Okay, so these are membrane-bound vesicular structures formed by the processing of packaging of Golgi apparatus. Now they are actually formed with the help of Golgi apparatus. And the thing which is special in lysosome is they are full of enzymes. Okay, full of enzymes they are. Okay, like different kinds of enzymes, lipases, proteases, hydrolases, everything they have. Okay, and what is the speciality in lysosomes is they actually help in digesting molecules. Okay, digesting different molecules. Okay, so it can be some food materials also, it can be some proteins, carbohydrates, lipids, nucleic acids, anything can be there. The other thing lysosomes do is they actually, if somehow some foreign particle, some bacterial particle or some genetic particle comes inside, some foreign particles, they come inside, they can catch them also and they can digest them and send it out. Okay, so foreign particles are also digested. Okay, so that digestion of foreign particles is known as phagocytosis. Okay, so that kind of function also is handled by lysosomes. Okay, so now when we talk about foreign particles or some viral particles, bacterial particles getting digested by lysosomes, that means what? Lysosomes are present in the immunity cells such as macrophages, they are there. Okay, macrophage does what? Macrophage, they eat up the foreign particles and they digest it out. That command macrophage. The lysosomes are the ones that digest the materials with the help of its different kind of enzymes. Okay. The last one is the vacuole. Now, vacuole, a big chunk of vacuole is present in the plant cells. Okay, big vacuole present in plant cells. Okay, now smaller ones are actually there present in this thing, animal cells, but it's not that important. Okay, small ones are present, smaller in animals. Okay, now in animals that actually they don't have a lot of function. Okay, it's the plant cells where they function a lot. So what is vacuole? It is a membrane bound space in the cytoplasm. It's just a space. Here you can see it's just one space. It doesn't have a lot of functions or anything. Basically it is kind of a storage material kind of a place. Okay, so they contain water, sap, excretory products, and other materials not useful for the cell. The basic thing when it comes to the basic function when it comes to vacuole is it is a storage area. Okay, now vacuole is actually bound by a single membrane called tonoplast. Okay, there is a membrane over it. Okay, that is known as tonoplast. Okay, and these vacuoles can actually occupy around 90% of the volume. Okay, so 90% of the plant cell, especially the plant cell, and is actually captured by the vacuole. So it's so huge. The rest of the organelles, such as mitochondria, nucleus, and chloroplast, everything, they live in the rest of the 10%, whereas the vacuole actually takes around 90% of the area. Okay. Now in plants, tonoplast, that is the outer membrane, it facilitates the transport of the number of ions and other materials against concentration gradients into vacuole. So different kinds of like molecules and everything is pushed by tonoplast into vacuole. Okay. In amoeba, the contractile vacuole is an important osmoregulatory function in many cells, like different kinds of cells, as I said, the food, okay, food particles, like plants store starch here. Okay, animals also store some kind of different food materials, although they are very small in nature in animals. Plants mostly store starch in here, apart from other molecules. So basically, vacuole is what they have the storage capacity because it's so huge. 90% of the cell, it's so huge, so they have the storage capacity in the cell. Okay. So that's it for today. In the next class we'll be discussing about the different kinds of other organelles. We'll discuss about the nucleus, we'll discuss about the chloroplast, and we'll discuss about the mitochondria. Okay, and little bit about centrioles and all. Okay, so now we'll discuss some little questions and then we can say goodnight to each other. Okay. Now, which part of the cell contain hydrolytic enzymes? As I said, okay, different kinds of hydrolytic enzymes, okay, hydrolases, okay, now other things, okay, these kind of enzymes are not present in flagella, centrosome, ribosome, they are present in lysosomes, okay. So, hydrolases and pepsinases and etc., they are present in lysosomes. What layer of protects every cell from its environment? Membrane, smooth ER, nuclear envelopes, cytosol. So it is membrane. I know. because it is apart from cell wall, okay, mostly in plant cell. But in animal cells, it should be cell membrane because they don't have the cell wall for protection. So the animal cells have cell membrane for protection. And passing of molecules, etc. Okay. Next, in the Golgi complex, the structure which is the functional unit is cisternae, thylakoid, mycoplasm, cristae. It is cisternae, right? The stack like this. Okay, they have a cis phase and a trans phase. Okay, so this is the trans phase and this is the cis phase. And these stacks are known as cisternae. Okay, so in Golgi complex, it is the cisternae. Okay, everyone, thank you for joining for today's class. If you have any doubts, you can put it in the chat box. Maybe in the next class, we can discuss about it. Okay, don't forget to like, share, and subscribe to our channel. Okay, and I'll see you guys in the next class. Bye bye. Good night.